Team Downward, Sustainable Future. Introducing teammates and mentors. Mentors are Forum Kupadia, Mithlesh Joshi, Lakshmi Garg. Teammates Kaushal Bhagat, Astha Patel, Fernandes, Murali, Jatin and Ravi. List of contents. Introduction to plastic pollution, polymer catalyst and sustainable solutions, application of enzymes, method to accelerate degradation rate, preparation of biodegradable polymers, life cycle assessment, and conclusion. Introduction. Polymer pollution primarily from plastic waste is growing environmental crisis impacting ecosystems worldwide. Traditionally, plastics which are made up from non-renewable fossil fuels do not break down naturally and can persist in the environment for hundreds of years. This accumulation of plastic waste harms marine life, wildlife, and even human health, as small plastic particles known as microplastic infiltrate soil, water bodies, and the food chain. Plastic pollute oceans, coastlines, and urban areas, creating a serious ecological imbalances that threatens biodiversity and damage natural habitats.
Polymers are integral to modern life, from packaging to medical and automobile uses. Due to their affordability and versatility, however, environmental impact is significant. Non-biodegradable plastics contribute to pollution, disrupt ecosystems, and harm wildlife, leading to urgent environmental issues like species extinctions. Our research focuses on sustainable solutions, particularly microbial degradation, enzymes such as hydrolysis breakdown to carbon structures of plastics. Facilitating decomposition, we also explore biodegradable alternatives like PLA, PHA, starch-based polymers already used in food packaging, agriculture, and medical applications like wound dressings. Accessing these materials through life cycle assessment ensure they are truly sustainable from production to disposal. Factors such as polymer structure and molecular weight affect how they degrade, with amorphous and lower weight polymers break down fast. Adopting biodegradable polymer is a medical, agriculture, and packaging sector could significantly reduce plastic waste and promote environmental health, paving the way of more sustainable future.
Catalysts that accelerate polymer degradation are crucial for reducing plastic pollution and promoting sustainability. Types of catalysts. Enzymatic catalysts like engineered PETs break down plastics like PET into monomers. Metal-based catalysts, for example cobalt and titanium, generate reactive oxygen species to degrade polymers. Photocatalysts like titanium oxide work under UV light to break down materials such as polyethylene. Mechanism of action. Catalysts lower the activation energy, producing reactive species that trigger polymer breakdown such as chain scission. Applications. Catalysts improve the efficiency of biodegradable plastics, assist in chemical recycling by breaking polymers into reusable monomers, and aid environmental cleanup by accelerating the breakdown of persistent plastic waste. Challenges. Issues include ensuring selectivity, efficiency under mild conditions, and producing non-toxic degradation products. With advanced sorting technologies like Fourier transform, infrared spectroscopy, recycling is becoming more optimized. Continued innovation in catalytic degradation and recycling technologies is key to building a sustainable circular economy.
Methods to accelerate the degradation rate of biodegradable polymers. There are mainly two methods. The first is abiotic method and second is biotic method. Biotic process occurs by use of living organisms like bacteria and fungi, while abiotic process occurs without direct involvement of living organisms. Hydrolysis, photolysis, oxidation are examples of abiotic degradation. Then increase the surface area. Crushing the polymer into micro or nanoparticles or forming a porous structures can increase the surface exposed for environmental interactions, which leading to a faster degradation. Mixes well with other materials. Polymer flexibility can be improved by adding specific substances such as plasticizers, which can lead faster degradation rate. By reducing the molecular weight, it's a simple. If polymer with lower molecular weight are more biodegradable because of their easier way to depolymerize. Then moving further, enzymes or microorganisms. If we treat polymer with enzymes which would decompose its chemical bond or microorganisms, example is PLA degrades quickly in the presence of certain microbes or fungus. The other is biostimulation. It's a method used to help bacteria break down pollutants by adding nutrients to environment. The other is bioaugmentation. Bioaugmentation is a technique that involves introducing microorganisms into contaminated areas to enhance its ability to remove pollutants. Then moving further, there are some environmental factors which affect the degradation rate. The first is humidity, the second is temperature, and the third is change in pH, and the other is reducing crystallinity. Less crystalline phase is more stable than the amorphous phase, so polymer with low crystallinity will usually degrade faster.
Now we will be having the application of enzyme in the degradation journey of biodegradable polymers. Enzymes are critical to the breakdown of biodegradable polymers as they speed up the process by catalyzing the hydrolysis of key chemical bonds like esters and amides. These enzymes include lipases, proteases, and specific depolymerases depending upon the substrate, and they work by first eroding the polymer surface and then as they penetrate deeper, initiating bulk degradation. This breaks the polymer chain into smaller, eco-friendly molecules that microbes in soil, water can further decompose. The efficiency of enzymatic degradation varies depending upon the polymer's structure, and polymer with the crystalline and hydrophobic properties are harder to break down, while the more amorphous and hydrophilic polymers are more accessible to enzymes. For instance, polyhydroxyalkanoates (PHA) are biodegradable by specific PHA depolymerases, whereas polylactic acid, also known as PLA, is effectively broken down to proteinases by K and certain lipases. To improve the efficiency, scientists are engineering enzymes to be stable in diverse conditions like extreme temperature and pH. For this, they are using thermoacidophils and salidophils as a bacteria, making them more practical for broader applications. Overall, enzyme-based degradation offers an eco-friendly solution by converting polymers into non-toxic residues, ultimately reducing plastic pollution and supporting our main goal, that is the biodegradable polymers and the sustainability of our resources. Now we will be having a look on the videos. This video basically demonstrates the biodegradable polymer. It's a very simple video. In this, we can see that biodegradable polymer can be prepared in a very simple manner, just by addition of a very simple substrate like glycerol, vinegar, which contains some acids, and the cornstarch, which is easily available in any of the laboratories. Now please have a look in the video.
The easiest way to make bioplastic is to combine 10 milliliters of distilled water, 1 milliliter of white vinegar, 1.5 grams of cornstarch, and 0.5 grams of glycerol in a pot and stir. Boil the mixture until it becomes clear and thick. Then pour it onto parchment paper in the shape you want. Let cool for 2 days or until fully hardened, then use.
Assessment of biodegradable polymers provides an in-depth analysis of the environmental impact these materials have over their entire lifespan. This method is useful for evaluating the sustainability of biodegradable polymers compared to traditional plastics, yielding wide material choices that minimize environmental harm. The LCA process for biodegradable polymers involves several stages. This would involve in the raw material extraction phase. Biodegradable polymers are generally sourced from renewable materials like corn, sugarcane, or starch, as opposed to fossil fuels. This stage examines the environmental effects of cultivating, harvesting, and processing these crops. considering factors such as land and water use, fertilizers requirements, and greenhouse gas emissions. The production and manufacturing stages evaluates the transformation of raw materials into polymers, focusing on energy consumption, emissions, water use, and waste generation. Although producing biodegradable polymers typically requires less energy than the petroleum-based plastics, it still emits some greenhouse gases and harmful substances. The LCA then assesses the total environmental impact by aggregating data from all stages to analyze the carbon footprint, water and air quality effects, soil health, microplastic risks, and the biodiversity changes due to agricultural practices or land user shifts. This helps determine if biodegradable polymers provide an environmental advantage over traditional plastics and highlights areas for improvement. Finally, LCA examines different disposal methods. Some biodegradable polymers can be composted, which returns nutrients to the soil. However, in landfills, these materials may not fully degrade and can release methane under anaerobic conditions. Incineration offers energy recovery but results in emissions. Recycling, though possible for some biodegradable polymers, requires carefully sorting to prevent contamination. The LCA assesses how each disposal option affects soil, water, and landfill conditions, as well as any potential for microplastics residues, providing a comprehensive view of the material's environmental footprint.
Conclusion. By working together and making conscious choices, we can create a healthy, just, and prosperous world for future generations. Let's commit to building a sustainable future now. Thank you.